Hello, it's Mr. Enzrud, and today we're going to be talking about Hardy-Weinberg and microevolution. So we're going to start off talking about genetic equilibrium. So genetic equilibrium is a situation in the real world where allele frequencies in a population remain constant. So that means that the percentage of alleles for a trait aren't drastically changing in any direction. This diagram just shows all the different possible alleles in this population of mice. So we have a certain percentage of black alleles, a certain percentage of brown alleles, and those allele frequencies aren't changing if they're in genetic equilibrium. So when we talk about genetic equilibrium, that really leads us to Hardy-Weinberg. So Hardy and Weinberg were some researchers that were really looking at debunking a myth about dominant alleles and how people commonly believed that the dominant allele in a population would increase in frequency until the recessive allele was gone. And so Hardy-Weinberg principle just says that equilibrium in a population, so those equilibrium of those alleles, allele frequencies, can only happen if a certain set of conditions are always true. And those conditions are that there are no mutations in a population, that there are no selective forces, so natural selection is not taking place, that the population is really large, that there's no movement in or out of the population, and that mating is random. And so in real life, these conditions are never met. It's not realistic. Some of these, if not all of these conditions, are happening in populations. But what Hardy and Weinberg determined is that these are the forces that actually cause evolution. And in the absence of these forces, so in a certain moment of time, like you look at a population right now, we don't really see mutations happening right now having an effect on a population, or the breeding or the movement of alleles in or out of a population. So if you look at a population a moment in time, then you can determine what those allele frequencies are. And we'll talk about Hardy and Weinberg a little bit later on. So what is microevolution? So microevolution is any change in the allele frequencies in the gene pool of a population. And so if we go back to Hardy-Weinberg conditions, we can really determine what causes microevolution, and that is mutation, natural selection, non-random mating or sexual selection, genetic drift, and gene flow. So those are the conditions that cause evolution to take place, or starting off with microevolution, changes in allele frequencies, which eventually leads to new species and evolution. All right, so what is a mutation? Well, mutation is a change in the DNA sequence, and mutations are the only way that we can get new alleles in a population's gene pool. So you can't have new alleles unless mutations happen, and mutations do happen. Now, most of the time, mutations are harmful, and if the cell can't fix the damages to the DNA, then it self-destructs through a process called apoptosis. So a lot of times, there's no impact on the mutation. Of course, sometimes mutations can lead to bad things like cancer, but every once in a while, mutations can cause new phenotypes. If you think about eye color, the pigment in your eyes, the brown pigment in eyes is melanin, and in the mutation that where people have blue eyes, there's a change in that protein which causes the brown pigment to be or to not be expressed, and so instead you see that blue color. So mutations are the only way we can get new alleles in a gene pool, and sometimes those mutations can be beneficial. Natural selection, we talked about in our last lesson, but natural selection is really the driving force of evolution. So when traits are selected by the environment, those that have a beneficial trait will survive, and then they'll pass that trait on to their offspring. So one really important key is that no one trait is always the best trait. It really depends on the environment. So the environment is what determines whether a trait is beneficial or not. If we think about the rock pocket mice again, um, whether the rock pocket mouse was black or brown um, didn't really determine its fitness unless you look at it on the backdrop. What did the ground look like? If it was that sandy soil, then the light-colored mouse was the beneficial trait because it blended in. But if it was the dark lava flow, then the black color was actually better because that mouse blended in. So it always depends on the environment, whether a trait is going to be beneficial or not, whether it increases fitness or not. Okay, so we're going to talk real quickly about 3 types of selection. And really this is like, what does selection do to a population? So the first type, stabilizing. Stabilizing selection is where the phenotype that's kind of in the middle of the population is the most beneficial. So being at either extreme, either higher or lower than that trait, is not beneficial. So stabilizing selection selects the average phenotype, whatever the trait is in the middle, and it selects against those extremes. A directional selection can move a phenotype from one type to another. So it moves a population from one phenotype to another. So this is where selection favors one extreme over another. And a great example of this is in Darwin's finches. In those finches, the beak shape changed depending on the food that was available. So birds with great big beaks in times of drought actually were benefited because they could go out and crack open nuts and seeds and use those as a food source. And then the last type of selection is called disruptive selection, and that's where 2 extremes are favored over the average phenotype. And this tends to lead to 2 different groups, creates 2 different groups, kind of the low extreme and the high extreme, and eventually that can actually lead to speciation, or the creation of different species. So those are the 3 types of natural selection and the effects that they have on the populations. All right, we're going to talk about genetic drift. So genetic drift is just a change in allele frequencies due to random chance. We have several examples of genetic drift here. One thing to keep in mind is that the larger a population is, the less affected it's going to be by these random changes. So my first example, just a random change. That big rock, it landed on some birds and it wiped out some of the birds. And what we're left with are the birds in the population that have the alleles that they have. So whatever birds are left, the alleles that they carry can be passed on in that population. And in this example, wasn't too big of an impact. The birds, the frequency of those alleles probably didn't change too much because there was good diversity and it was a nice big population. Now another type of genetic drift is called the founder effect. And this is when a small number of individuals breaks off from an original population and starts their own population. So here again we see these birds, and let's say 2 of these birds come over to a new island and they start a new population. Well, those birds that started the new population were only bluebirds, so they only had certain alleles that they were carrying with them. And so the offspring that they produced could only have those alleles because they didn't have any of the red-colored alleles brought with them. And another example of genetic drift is called the bottleneck effect. And this is when we have a drastic reduction in population size. So let's say these birds were introduced to a predator. So this big mean cat starts eating these birds, but it seems to prefer the red birds over the blue birds. And so the red bird size the population was kind of decimated, and we're left with mostly the bluebirds. So this is going to have a big impact on the allele frequencies in that population. A lot more of the blue alleles are going to be present than the red alleles. And finally, gene flow. So gene flow is the movement of alleles into or out of a population caused by interbreeding. So when a population, specifically when populations are separated by some type of land mass or water or something, they're not commonly interbreeding populations. And when we have the introduction of one of these species to the other population, then we're introducing new genes. So gene flow is the movement of alleles or genes to a population caused by this interbreeding. And lastly, we're going to talk about non-random mating. So non-random mating is also called sexual selection. This is when a mate chooses its mate based on phenotypic differences. This is really common in birds, especially the birds of paradise. These birds can choose their mates based on a variety of kind of wild traits, like feather coloration, or an elaborate song that their mate could sing, or wild, crazy plumage, as you can see in some of these pictures, or even fancy nest building, sometimes little mating dances that the birds will do. So non-random mating really confused Darwin because it actually creates an exaggeration of phenotypes because those birds are selecting for these wild, crazy traits. So finally, I'm going to finish up, just bring back Hardy-Weinberg. Hardy-Weinberg principle is explained with a couple of mathematical equations. P plus Q equals 1, and P squared plus 2PQ plus Q squared equals 1. Now, we're not going to get into those right now, but what these do is they help scientists predict the genotype and allele frequencies in a population. And so in our next video, we'll discuss how to use Hardy-Weinberg equations to predict those frequencies. I hope this lesson was helpful, and we'll see you next time.